All right, part 2. All right, so that was anaerobic. Let's move on to aerobic. This is the biggie here. So GLKEC, good luck kicking or kissing every cat. All right, cats are really squirrely. Good luck kicking all of them or kissing all of them. Cool. So we already did glycolysis. That happens in the cytoplasm. Everything else here, steps 2 through 5, are all going to be in the mitochondria. A couple different parts of the mitochondria, though. So this is a potential extended answer question to draw and label the mitochondrion, which is the singular mitochondria. But if you screw up that plural singular, that's not a big deal. So there are 1, 2, 3, 4, 5 parts that you need to know. We have the two membranes, the outer membrane, that's the smooth oval, and the inner membrane. Notice there's no T in here because it's not intermembrane, it's inner, like inside, like inner piece. So notice the inner membrane has these squigglies. It's not a smooth oval, it has these little squigglies in here. Those squigglies are called cristae. That's the plural. The singular would be one crista. So if you know any squiggly people named Krista, that's a pretty easy way to remember that. And then the space between this red arrow, outer membrane, is pointing to this black line. Inner membrane, also black line. Cristae, also these black lines. But the last two parts are open spaces. One space is really big, and that's the matrix. So I just think of the Matrix movies and how big the matrix actually is, the computer simulation, spoilers. And then the this one does have a T in it. Intermembrane space is the space between the two membranes. It's a lot of syllables, but the name is exactly what it is. Hopefully you're familiar with that prefix, like interstate. Inter means between states in that case. Intermembrane is literally just the space between membranes. Okay. Some important terms first. A little reminder here, the sugars are going to be oxidized in every single step. We are on steps 2 through 5 now. So that's LKEC. Every single step, the sugars are going to be oxidized because we need to reduce this low energy, kind of useless NAD. I mean, not useless. We need it to make NADH. We need to reduce the NAD into NADH, which now NADH is the high energy charged battery form that can power the last two steps in the mitochondria. Okay, we also got FADH2, which is just treated the exact same as NADH. It's just like the cousin. It's a little bit lower energy, but it's pretty good. This is a synonym you need to know. A lot of times you'll see the word reduced NAD. That is NADH, because when you take NAD and you reduce it, remember that the HO table, we can add an H and make NADH. Another new term is decarboxylation. So D means remove or, leave it out. So decarboxylation is losing CO2. When you do photosynthesis, you'll have carboxylation because you're gaining CO2 as the plant makes it into sugar. And H plus is literally just a proton, right? If we had H neutral, covering up this little plus sign here, H neutral, that is the smallest atom. That's a proton and an electron. So if we lose the electron and make the ion, now all that's left is a proton. Okay. Kind of a fancy term at the end here that combines two ideas. A gradient is a difference. So an electrical gradient is when you have different charges across a membrane. So like batteries have an electrical gradient. That is literally a synonym for potential or voltage. We saw this with neurons. They have an electrical gradient, right? Negative on the inside, positive on the outside when at resting. Chemical gradients is the more classical gradient that you probably think of. This is what drives diffusion and osmosis. That is our normal concentration gradient. A chemical is literally any molecule. So it's a very general term. And so if we combine those two, we get the idea of an electrochemical gradient. That would be both. We have a charge imbalance, and there's some kind of concentrated chemical that's more concentrated on one side than the other. Okay, so the easiest way to do this, create a chemical and electrical imbalance, is if we just have a bunch of ions, because ions satisfy those two requirements. Ions are chemicals. They're also electrically charged. So if we pack a bunch of ions into a small space and make a high concentration, we've just made an electrochemical gradient. And that ion, Ooh, there's an ion on this page. That ion is H plus. So the way that you make most of your ATP, we're going to pack H plus, make it very unhappy, pack it into a tiny space, and that's going to create an electrochemical gradient where it wants to go run out the door, and the door is a very important enzyme. The H plus wants to run out the door and leave its packed in space. Okay, actually, I'll introduce that idea right now. The packed in space is going to be the intermembrane space, and it wants to leave that packed in space and go into the free range, wide open matrix. Okay. All right, so we did G, again, GLKEC. We did G, that was glycolysis. Now we have some pyruvates, represented here as 3 carbons. Yeah, there's some oxygen and hydrogen in there, but we're going to ignore those. So the link reaction, we've done glycolysis. Now we have to take the pyruvate into the matrix. So we have to cross 2 membranes to get here, the outer and the inner membrane, to get into the matrix of the mitochondria. Okay, the matrix is the big open space in the middle. Matrix middle. Maybe that's a better way to remember. Matrix in the middle. So we're going to take the pyruvate. We're going to energize a low energy NAD plus. I left off the plus sign here because I got a little lazy. We're also going to use this random little molecule called coenzyme A. There's an enzyme here, and this is near an enzyme, so it's kind of a weird name. It's called a coenzyme. It has other uses, but anyway, it's just going to act as like a little shuttle here. So there's the the coenzyme A. It's going to be involved here, and the pyruvate is going to turn into acetyl-CoA. Acetyl has 2 carbons in it. Just like acetic acid is vinegar, that actually has 2 carbons in it. So as we lose the carbon, we're going to oxidize pyruvate. It's going to gain some hydrogens, lose some oxygens. And as it gets oxidized, we're going to reduce the NAD into NADH. Okay, so we've made something useful, and we've lost the CO2 from this carbon compound, right? We started from 6 carbon glucose, now we're down to 3 carbon pyruvate, now we're down to 2 carbon acetyl-CoA, 6 to 3 to 2. We are on our way to all single carbon dioxides. Okay, NADH is useful. That's going to power the ETC, which is step 4, GLKEC. And the CO2 is going to be breathed out. Okay, so overall, some vocab words here. Pyruvate is decarboxylated as it loses this CO2. It's also going to be oxidized because we need to reduce the NAD, right? You can't have a reduction without an oxidation and vice versa. So we're oxidizing the pyruvate so that we can reduce the NAD into NADH. Okay, now it's K of GLKEC. We have acetyl-CoA, which I'll remind you is a 2 carbon compound. So we still have some carbon-carbon bonds to deal with that we can extract some energy from and make CO2. So that's from the link reaction. The product of the previous step becomes the reactant for the next step. It's going to stay in the matrix. Steps 2 and 3 are both in the matrix of the mitochondria. So acetyl-CoA is going to be decarboxylated twice. We have 2 carbons here. We can lose 2 CO2s. We're also going to oxidize, which we did in steps 1, 2, and 3. We're oxidizing the sugar over and over and over. Okay, the products per acetyl-CoA. This is per acetyl-CoA. We get 1, I think of it as 113. I usually ignore the 2 CO2s because I can remember that acetyl-CoA has 2 carbons. So yeah, you're going to make these 2 waste products. In terms of the high energy carriers, I just remember 113, 1 ATP, 1 FADH2. And look, we make more NADH than anything else. Okay, this is going to power the next step. So these first 3 steps, we've made a lot of NADH, more NADH than anything else. Okay, you don't need the names of these compounds. You need to know a 2 carbon comes in. That is going to combine with a 4 carbon. Okay, so this 2 carbon combines with this 4 carbon. They come together and make a 6 carbon. And then that 6 carbon is going to lose 1, 2 CO2s that turn back into a 4 carbon. As it turns back into a 4 carbon, it's going to make 1, 2, 3 NADHs and 1 FADH2. So there's all the products there. Okay, ATP is used to power a billion different things. CO2 is breathed out. FADH2 and NADH is really what we were after here. So yeah, we made a bunch of stuff. all I really, really care about, yeah, ATP is useful, but these were the things I'm after. Important thing to note, I have seen this as a potential test question. This is per acetyl-CoA 113. So you would have to double all these numbers if we're talking about per glucose. Because for every glucose, I'll go back here to remind you, for every glucose, we get 2 pyruvates. Okay, so every time we want to do this cycle, we need a single acetyl-CoA, but per glucose, we got 2 acetyl-CoAs. Okay, that's not what this 2 stands for here. This means each acetyl-CoA has 2 carbons. So if you run this cycle twice, that's what you can get per glucose. So per glucose, you get 2 ATPs, 4 CO2s, 2 FADH2s, and 6 NADHs. Okay, so you would double it per glucose because you get 2 acetyl-CoAs per glucose. Okay, potential. Here's a test question from an IB test. So what is happening? Oxidized NAD plus? Nope, that's not a thing. It would be reduced NAD plus. The chemical energy stored is used to there we go, reduce that. Ooh, that one looks pretty good. That is exactly what's happening. Answer is B. All right, step 4, the big payoff phase. Well, I guess 5 is the bigger payoff phase. Anyway, so all the NADH and FADH2 we got from the previous steps. So per pyruvate, we get 1 from glycolysis, because that overall was 2, but we're cutting all these numbers in half because this is per pyruvate. So that would be 1 from glycolysis, 1 from link, and 3 from Krebs. And we got one FADH2 from Krebs. So per glucose, you would double all these numbers. So per glucose, we got 2, 2, 6. So that's a total of 10 NADHs and 2 FADH2s. So those are our big electron carriers. Remember, those are the high energy carriers. A little bit different to ATP, which is also an energy carrier. ATP powers stuff by giving up a phosphate. These 2 power things by giving up high energy electrons. And those electrons are going to power the ETC. So the ETC, in 2 words, is proton pumps. They are pumps. So those are proteins that do active transport and force things from low to high against their will. Also got some carriers to help shuttle the electrons around. So there are a bunch of pumps in the cristae, which was the intermembrane squiggly folds. And a proton, I'll remind you, is an H+. So these pumps need energy from somewhere. A little bit different from the sodium-potassium pump in your brain and throughout your body. The sodium-potassium pump uses ATP to power itself. These pumps use the electrons from NADH and FADH2. So they're going to use those high-energy electrons to be powered. And as they're powered, they grab H+ from their nice, happy home in the big open matrix, and they're going to pump them across the cristae, across the inner membrane, into the small intermembrane space. So we are packing in these protons. They were happy and free range and roaming around, but now we're going to pack them into the smaller space and make them unhappy. Okay, so electrons are going to be passed by carriers. Okay. This packing of H+ in the intermembrane space creates an electrochemical gradient. The H pluses are really unhappy. There's an electrical gradient because there's a buildup of positive electrical charge. There's a chemical gradient because this is, kind of a chemical. This is an ion, so they want to go from high to low, right? They want to rush back into the matrix because of an electrical reason. They want to get away from their positive neighbors, and a chemical reason. They want to spread out and not be so highly concentrated. Okay, last part of the ETC you need to know is that electron needs somewhere to go. Okay, in this picture here, the electrons are donated by NADH and FADH2. These blue blobs here are the pumps. The arrows are showing H pluses being pumped from the matrix down here into the small intermembrane space. So this phospholipid bilayer is the outer membrane. This is the inner membrane. It doesn't show any folds here, but there would be squiggles in this inner membrane, maybe if we went farther to the right or left. Anyway, so these are the pumps. This is the ETC. And so the electrons are going to go left to right, powering these pumps. And the problem is, as they power the pumps, the electrons lose energy, and now we have a useless low-energy electron. it needs somewhere to go. Electrons need a home. So that home is going to be oxygen. Oxygen sounds like such a big deal, but its function is so simple. It grabs the low energy electron to give it somewhere to go. It is the terminal electron acceptor, TEA. It's the T. Let's spill the tea about oxygen. So terminal means it's the final, it's the end resting point of the electron, and it's accepting the electron. Okay. And then so that oxygen, as it grabs electrons, it needs to turn into something. So it's also going to grab some H's and turn into H2O. Okay. And you might be like, wow, oxygen got used so late and for such a seemingly small job, but this is a huge job. If there's no oxygen, the low energy electrons get stuck in the pumps. These pumps become worthless with their being filled with low energy electrons that have nowhere else to go. And so NADH and FADH2 are stuck. They can't give up their electrons because there's already electrons, useless low energy electrons in the pumps. So NADH would need somewhere else to go. And so that was the whole purpose of fermentation. NADH needs to turn back into NAD+. Ideally, it would use its energy for good to power these pumps. But if these pumps are worthless because the electron had nowhere to go because you didn't have oxygen, then NADH needs to give up the electrons somewhere else. And so it'll give it up to fermentation, to pyruvate, to make our waste product. What a waste of energy. But we had to regenerate the NAD somehow. Okay, so the overall product of the ETC, this is the icing. This is the cake and the icing on the cake. We have a buildup of H plus. That is a very, very big deal, very important. We've also made our NAD and FAD, which can then go back into steps 1, 2, and 3 and be re-reduced back into their high energy compounds. So this is these are low energy, but they're still useful starting materials for making their high energy counterparts. And then water, not a big deal. You have water everywhere in your body. You don't even notice this water. Okay. Hibernators notice this water, but not you. Okay, so here's a picture from an IB test. Where do glycolysis and the ETC occur? It's just called electron transport, but they mean the chain. So glycolysis is in the cytoplasm, so that would be S. What we're looking at here is one mitochondrion. These might be vesicles or vacuoles. It's kind of hard to tell from the picture what these other little spheres of membrane are. And then the ETC occurs along the cristae. So it's kind of hard to see what's pointing at what. I think Q is pointing at the inner membrane where those pumps would be chilling. Cool. All right, last step, chemiosmosis, GLKEC. Chemi means chemical, literally any molecule. Osmosis is the passive transport of water. I kind of hate this term. It's really stupid. Okay, it literally just means diffusion. It's like, why not just call it diffusion? No, because we need a fancy sounding thing. Okay, it's a chemical doing passive transport, but I thought osmosis was only of water. Yeah, usually, but it's a stupid word. Anyway, so what we have is a high concentration of H plus that we've made from the first 4 steps. Now we're going to harness that power. So imagine water up behind a dam on the top of a hill. If we open that dam, that water can rush out with a lot of energy. That's potential energy. That is the situation we have here. We have a lot of unhappy H pluses. They want to leave that small intermembrane space. This would be the intermembrane, this is hard to do, space. Okay, these H pluses are unhappy in this intermembrane space. Okay, so the only way they're allowed to actually leave, they can't just fit through the bilayer because they're charged. And these fatty acid tails are non-polar, so they would repel the charged polar stuff. So the only way they're allowed through is through this very, very special enzyme, ATP synthase. Look how complicated that guy looks. As the H plus rushes through, this thing literally spins. It's really cool looking. As the H plus rushes through, the ATP synthase, it spins the ATP synthase. ACE means enzyme, so it's a big old protein. And as it spins, it pushes a phosphate and an ADP together to make ATP. Okay, so all of this process was just so we could spin, the previous 5 steps are just so we can make this thing spin and push the phosphates together with an ADP. push one phosphate with an ADP to make ATP. The subscript I stands for inorganic. Um, I don't know why it needs to be there. All phosphate is inorganic. That's weird. 90% of your ATP is made by ATP synthase. Makes sense with the name. To synthesize something is to make it. We are making ATP. Okay, so this is phosphorylation because we are adding a phosphate to ADP. Okay, useful YouTube video to watch this whole process. I'm not going to go through it now, though. Okay, so the whole purpose, let's take a step back. The whole purpose of cell respiration, we were decarboxylating, removing CO2s, oxidizing sugars so we could reduce these low energy compounds into high energy NADH and FADH2. And so those things could give up their high energy electrons to power the ETC. Okay, powering the ETC creates an H plus gradient. The H plus gradient rushes out, powers ATP synthase. That's a lot of intermediate steps. Okay, a very important vocab word. This could be an extended answer 8-point question in 3 words. Describe oxidative phosphorylation. Steps 4 and 5 together, the ETC eventually powering chemiosmosis is called oxidative phosphorylation. Oxidative is step 4 because NADH and FADH2 are being oxidized as they lose their electrons. Remember, Leo, losing electrons is oxidizing. That powers the ETC. Phosphorylation is step 5, where the ATP synthase is powered by the H plus rush. As the H plus rushes out, the ATP synthase puts the ADPs together with the phosphates to make ATPs. Okay. Structure function of mitochondria. The reason we have these squiggles, 3 important parts. We have a large open matrix for steps 2 and 3, Krebs cycle and link. Sorry, that was the opposite order. Link reaction and Krebs cycle take place in the matrix. We don't need any membrane for those two. It was just pyruvate being decarboxylated and acetyl-CoA being decarboxylated and oxidized. So we just need enzymes. We didn't need any pumps or membranes for that. So that's floating around. We also want the large open matrix so that the H plus can spread out and be happy there and want to be there. Okay, then we have these folds, the cristae. We fold the inner membrane to give it more surface area. If it was a smooth oval, that would be less membrane. So we put these folds in there. That's a lot of membrane to increase surface area. So we can throw in a bunch of pumps. And remember, those pumps are the ETC. That's all the ETC is. It's just a bunch of proton pumps. H plus is protons. We also use the cristae for step 5. The ATP synthases are also sitting on this black line, just like the pumps. So you'd have, a billion of each of these for those last 2 steps. We also want a small space so we can make the H plus unhappy. Remember, the pumps are going to pump the H plus this way, make them unhappy, and then the ATP synthase lets the H plus rush out this way, right? It's just like moving water uphill to give it some potential energy, and then letting the water rush downhill through a dam to power the turbines and generate electricity. Okay. Big sum up table. So if none of that made sense, memorize this table. Okay, locations. Cytoplasm for step 1. This is steps 1, 2, 3, 4, 5. GLKEC. Cytoplasm. Matrix of the mitochondria for steps 2 and 3. No membranes needed, just big open spaces. Electron transport chain. We're going to pump the H plus from the big happy matrix into the small intermembrane space. Step 5, chemiosmosis. We're going to let the H plus rush back into the matrix from where it came. And as the H plus rushes back through, we're going to make a lot of ATP. So for the carbons, 6 carbon gets split into 2 3 carbons. Those 3 carbon pyruvates are decarboxylated once and then decarboxylated twice, all the way down into CO2s. Okay, we make NADHs in all 3 of these first steps to be used in the 4th step. Okay, we make a bunch of, the only time we make CO2 is actually in the matrix. The reason you need to breathe out is because of what gets made in the matrix. Okay? A nice little way to review this that I like to do is to talk through this table, but in reverse order. So you can be like, okay, the whole purpose of cell respiration is to make a lot of ATP. Where did it come from? It came from an H plus gradient. Where did the H plus gradient come from? It came from oxidizing the NADH. Well, where did the NADH came from? It came from oxidizing sugars. Okay, so that's kind of a big sum up going from backwards. Oh, a nice little song here. Maybe I'll sing that later for you if you're lucky. Okay, and then some good questions to go through here. Cool.